assistant professor in the Department of Earth Systems Science, Microbiology, and she got her start in her college career as a bachelor's graduate from Harvard University. At that time, if I'm not mistaken, she had an interest in space science and did work. You might have even isolated a bacterium from a spacecraft or something like that. I don't know. After that, she worked for a while at the Jet Propulsion Lab in California, and that was before graduate school. Graduate school was Caltech for her PhD in the study of the microbiota, in particular bacteria, Archaea, in deep sea sediments. After completing that work, if I'm not mistaken, I better check my notes, there was a stint at Lawrence Livermore Laboratory. She's been to a great many popular and famous campuses, from Harvard to Caltech to Lawrence Livermore, and now finally you have a football team to root for, because she's at Stanford University, and her interests are in deep sea biology, the real deep sea, and she's been studying nitrogen fixation, methane oxidation, and other interesting and important microbial processes in the deep sea as they affect the global climate through deep sea biology. And she's going to talk today about a particularly innovative, I think, and exciting topic, and that is analyzing and interrogating the metabolism of organisms in the deep sea using mass spectrometry at the single cell level. So we'll give a warm hand for Ann Dekas, and her talk is a single cell view of microbial activity in the deep sea.
Yeah. Thank you for that very nice introduction, and thanks to all of you for coming out today. It's really a pleasure to be here. I'm going to talk a little bit about microbial life in the deep ocean and why I think it's important to study it at the particular scale that I do, which is on the single cell level. So I call myself a geomicrobiologist, and kind of very broadly what I'm interested in is understanding the role of bacteria and archaea in biogeochemical cycles over long time periods, so the history of the Earth and potentially the future of the Earth. And I'm interested in this for a few reasons. One is to explain climatic events in the history of the Earth. One is the search for life on other planets. I've been interested in astrobiology for a long time, and while we'll probably get to very few extraterrestrial sites in my lifetime, I think our ability to remotely detect the chemistry of other atmospheres is a lot more likely. So understanding how microbes are shaping the chemistry of those planets can help us do this remote detection of life. And predicting future changes on Earth's climate. Future changes in Earth's climate. So the specific research questions I end up asking are, who does what in situ? So we have these long lists of the species in a particular location, but what are they actually doing? What influences who does what and how much? So the geochemical interaction with the biology. And will these metabolisms act as a positive or negative feedback to climate change? As our world changes, how will the organisms within it change? So one of the big challenges in asking questions like this is bridging a whole range of spatial scales. On one end of the spectrum, we have specific individual molecules like DNA, and on the complete other side, we have the entire world. And we're trying to expand up to understand these tiny individuals and how they're having an effect on the entire Earth. And there's different types of science that focus on these different areas of this spatial scale. We have modelers that are primarily doing the big side of it, molecular biologists who are primarily doing the smaller side of it. Geochemists often come in handy here when you're looking at the flux of particular chemical species in and out of a habitat. But one of the ranges of this spatial scale that's been a little bit understudied is the scale when you're looking at individual cells and how these interactions on the scale that kind of matter to the bacteria and archaea influence their activity. So if we were to look at the entire ocean water column, we'd see microbial cells distributed kind of like this. About 30% in the top sunlit area where the vast majority of the research happens, But then the majority of the cells in the dark middle of the water column where much less research happens. And then about 15% of the cells in the top 10 centimeters of the sediments alone. So from this alone, you should get the idea that the sediments contain extremely densely packed microbial communities. In each of these zones, you have different chemical regimes which result in different metabolic activities. So at the very top, you have both light and oxygen. In the middle, you have darkness, but you still have oxygen. And then in the sediments, you have darkness and very rapidly no oxygen. So the different metabolisms you have in each of these depths have different implications for greenhouse gas cycling. So I've drawn a couple of these up here, focusing on CO2, nitrous oxide, and methane cycling. Some of the work that I'm going to talk about today relates to nitrogen fixation in deep sea sediments, and this is going to be linked with methanotrophy at methane seeps. And then we're going to get into the activity and metabolic flexibility of marine archaea. So all the things we're going to talk about today happen in the dark, deep part of the ocean. We're not going to be talking about the photic zone. So one of the big problems with studying environmental microbiology in general is that the vast majority of microbes have not been cultured. What this means is that the things that we know a whole lot about based on traditional microbiology experiments don't tell us that much about what is actually going on in the environment where we have the full diversity of organisms. The solution to this is to study environmental samples where we have the full diversity of organisms present and to use culture-independent techniques. So that's primarily what I'm going to be talking about today, and particularly the single-cell approach using nanosims. So stable isotope probing is a kind of general technique which can take many different forms. So the basic concept here is that you have a mixed community of cells. You don't know who is who. It could be all different species. If you take the isotopic composition of these cells, you'll get a natural abundance ratio, for instance, 15N to 14N, is in the environment at a pretty predictable ratio in most places. If you were to incubate those cells with an isotopically labeled substrate, like 15N2, you would see if any of those organisms were capable of eating and assimilating that chemical species and actually putting that 15N into their DNA and their other biomolecules. Then when you take their isotope ratio, it's going to be above the natural abundance, and you have an indication that they were capable of doing that activity. So you can measure this a few different ways. One is to take a bulk isotope measurement of an entire community. Another is to extract the DNA from this sample, and based on the tiny increase in density you get from incorporating that 15N instead of 14N, you can separate out the DNA of all of the active community members in a density gradient during a very long centrifuge step. So you have all of the 13C labeled DNA down here, and the 12C up here, same concept with 15N and 14N. And then you can sequence all of these different fractions to figure out who, based on the DNA sequence, was doing that activity. So in this sense, you're actually linking who it was to what they were doing. And then the final way, which is the way I'm going to be focusing today, is nanoscale secondary ion mass spectrometry. And in this way, you're getting an individual isotopic measurement of a single cell. So a nanosims is, if you've seen Honey, I Shrunk the Kids, it looks a lot like that instrument. It's quite large, and it has a lot of wires hanging off of them. There's about 7 in this country, at least, that are available for biological analyses. And one of them is at Stanford, and one of them is at Livermore. The analyses that I'm going to be talking about today were done at Lawrence Livermore National Lab. You prepare your sample a lot the same way that you would for an SEM analysis, so it's dry and in a vacuum. You hit your sample with a primary cesium or oxygen beam. That beam has a diameter of about 50 nanometers, which is what gives it this really high spatial resolution. It rasters over the sample like an old-fashioned TV and puts together an actual image, an isotope image of your sample. If you're collecting these secondary ions, you can collect up to 7 at once. So you get an image like this, where each one of these little blobs is an individual bacterial cell or something else. We don't really know who is who or what they are. So how are we going to link that? We do another procedure upstream of this called fluorescence in situ hybridization, where we actually add fluorescently labeled probes to particular species, which turns them different colors. Very convenient. So now when we look through the microscope, we actually know which species are there, and then we can put those in the nanosims. There's our Honey, I Shrunk the Kids machine. And then you can link which species it was to what it's actually doing. So here's an example of the microscope fish image where this particular green cell is the one we're interested in, and the element and isotope image from the nanosims where we can see that same cell and get information about what it's actually doing. So coming back to the scientific questions we're after, we're going to start with nitrogen fixation at methane seeps. So nitrogen fixation is microbial metabolism where you're fixing N2 gas into ammonia, which is then used for biomass. All life needs nitrogen. Nitrogen gas is the biggest source of nitrogen on the surface of the planet. And the organisms that are capable of assimilating nitrogen gas kind of unlock this reservoir of nitrogen for the rest of the ecosystem. Not all organisms are capable of nitrogen fixation. The subset that are called diazotrophs. And what's neat about this process is that it's a very important process for industry as well. The Haber-Bosch process, if you've heard of it, is what produces fixed nitrogen for our fertilizer. And that process requires extremely high temperature and pressure in order to break this stable triple bond. And biology is able to do this at ambient temperature and pressure using the enzyme nitrogenase. So it's very neat, I think, that bacteria have solved this process in a very elegant way when humans have to put in so much effort to do it. It's not completely free for bacteria either, though. It takes a big input of energy for them, too, in the form of ATP. So these organisms only do it if they have to do it, if there's no other sources of nitrogen available. So it plays the role in an ecosystem of overcoming nitrogen limitation, because when there's no other nitrogen sources and everybody is stuck, the organisms that are capable of fixing this nitrogen gas get started and then provide nitrogen for the rest of the community. One of the reasons I got interested in looking at nitrogen fixation is because there is a debate over whether or not the nitrogen cycle is balanced on the global marine scale. And so about starting maybe 15 years ago, people started to notice that the rates of denitrification exceeded the known rates of nitrogen fixation by about 2 times. But if you look at longer scale records in the rock record, you see that there shouldn't actually be an imbalance of this nitrogen cycle. So it seems like instead of there being an imbalance, our inventories are incorrect. And particularly that nitrogen fixation was being underestimated. So we started to wonder, could nitrogen fixation in sediments be one of these underestimated sources of fixed nitrogen in the marine environment? So marine sediments cover about 2/3 of the entire planet. You guys know here, since you are ocean scientists, what a big fraction of the entire Earth the ocean is. And I've already mentioned the cell density in sediments is about 1,000 times greater than that in surface water. So there's a ton of metabolic potential in the organisms that live there. They host really critical metabolisms, including CO2 and methane cycling. But they're not thought to host nitrogen fixation because there should be a lot of other reduced nitrogen sources in sediments just from the breakdown of organic matter. So previously, people thought no nitrogen fixation was probably happening in these locations. The weird thing, though, was we kept discovering the NIF genes. So NIF genes encode for nitrogenase, this enzyme which mediates nitrogen fixation. So if we're seeing this gene everywhere on the seafloor, doesn't that suggest that maybe this process is happening? So we wanted to look into it. Our questions here were, are these deep sea diazotrophs actually functional? Who are they? What are the environmental controls on this process? And is nitrogen fixation actually an important source of bioavailable nitrogen in these sediments? So we sampled at a series of places all along the west coast of North America, and we were targeting places where we thought nitrogen limitation might be an issue. And places where it might be an issue are primarily places where there's tons of carbon loading. A lot of places in the deep sea are limited by carbon, but if there's plenty of carbon, you might be limited by nitrogen. So we went to methane seeps. We visited them with submersibles, so either manned or remotely operated submersibles to collect the samples. We divided the cores that we collected into different depth horizons. We incubated them with 15N2 because we're interested in this nitrogen fixation metabolism, but we also incubated them with 15N ammonium, which is a proxy for overall growth. So basically all organisms can, or microorganisms can take up ammonium. So this is just telling us who is active in total. And we incubated with both argon and methane. Then we analyzed it with fish nanosims, as I've already explained. We sequenced the DNA, and we also did bulk measurements of how much nitrogen fixation was happening total. So I have a little video to give you an idea of what it looks like to collect these samples. This is background seafloor. This is at Hydrate Ridge off the coast of Oregon. Many of you might be familiar with this marine snow that is raining down. This is quite different from the methane seeps, where you actually have bubbles of methane ascending from the floor. This is naturally occurring. It happens along continental margins around the world. You can also see lots of carbonate rock, which precipitates in situ as a result of this methanotrophic metabolism that changes the chemistry of the water. Here you can see a core being collected in a sulfide oxidizing bacterial mat. The sulfide oxidizing bacteria grow where methane is seeping beneath because the methane is oxidized with sulfate, which produces sulfide, which then supports those sulfide oxidizers on top. So I think I'll stop that there. So the organisms that are actually taking advantage of this methane are anaerobic methanotrophic archaea, which are here in pink because of the fish probe that I've added, and sulfate-reducing bacteria, which are here in green. This is an obligate symbiosis. You see them in really amazing associations with each other. And they are found at methane seeps around the world, and they're thought to consume about 80% of the methane before it reaches the water. So they are a pretty efficient methane filter in the sediments. After we incubated these organisms with 15N2 for 6 months, we put them in the nanosims, and we were able to see, based on the 15N14N ratio, that they were, in fact, consuming this 15N2, and the primary consumers were the archaea in the center of the consortia. So here, black is natural abundance, so anywhere where you're seeing a color is enriched in 15N, so we think the archaea were consuming it and then also sharing it with the bacterial partner. And we also had sequences that we recovered from the same sediments that showed that the NIF genes were in the same order as they are found in diisotrophic methanogens that are found in pure culture. So once we saw this in one location, we sampled methane seeps from several different locations and repeated it over and over again. And what we saw, in addition to just seeing that nitrogen fixation was happening, was that where we saw the number of these methanotrophic aggregates increasing with depth corresponded to the peaks of nitrogen fixation we saw. We did not see nitrogen fixation everywhere. We saw it in very discrete depth horizons, which suggested that either the geochemistry or the distribution of microorganisms was dictating this. And it seemed to overlap with where we had the most methanotrophic archaea. When we did incubations with and without methane, we also saw that nitrogen fixation only occurred with methane. So based on these two pieces of evidence, the overlapping distribution with the methanotrophic archaea and the lack of nitrogen fixation without methane made us think that these methanotrophic archaea were the primary nitrogen fixers in these sediments. The funny thing, though, was that the NIF-H sequences we discovered were actually extremely diverse. So even though the archaea seemed to be the primary nitrogen fixers, they weren't the only ones. You can see that in this pattern here. So in black are NIF-H sequences that are representative of the whole diversity of NIF-H sequences in any habitat. They are usually classified as these different clusters, 1 through 4. The red sequences are the ones we found at methane seeps, and they're distributed kind of throughout this diversity. So the archaeal ones are here, but then we're finding them all over the rest of the tree too. And in this site, which was a methane seep in Costa Rica, we're looking at the transcripts, not just the DNA, so they're actually being expressed. So there must be a diversity of diazotrophs mediating this at methane seeps. We tried to look into that a little bit more by using a variety of different fish probes to target different organisms at seeps. And one of the groups we tried to target was a few different sulfate-reducing bacteria. And one of the interesting things about sulfate-reducing bacteria at seeps is that they're found in two spatial associations. So one is clustered with the methanotrophic archaea, like I showed you a picture of. And then the other way they're found is just free living. So using the nanosims, we could separate, we could independently analyze the ones that were attached to the archaea versus the ones that are free living. These are different scales here. So this is like 1 micron, and this is 10 microns here. And what we saw was very different activity patterns when they were attached to the archaea and when they were not attached. And what we learned was they were 15N enriched only when they were attached to the archaea and not when they were separated, which told us they really weren't independently doing it as separate cells. So our next step was moving outside of methane seeps to look at another place where there was carbon loading, but a different type of carbon loading. So we looked at whale falls. So many of you might be aware, whale falls are places where whales' carcasses have sunk to the bottom of the seafloor. This happens naturally, but it also happens with the help of the Coast Guard and MBARI when you want to study them for scientific use. So this particular one was natural, but two of the other ones we looked at were kind of arranged. What we were wondering there was, if you add complex organic material, so carbon and nitrogen, that's in whale biomass, do you still see nitrogen fixation? I'm showing you 5 different cores here. This one is a whale, this one is a whale, this is like 50 meters away from that whale, and this one is 50 meters away from this whale. And this is just the pore water ammonium concentrations. We did see nitrogen fixation at each of these sites, and again, we saw it in very discrete spatial horizons. So the dark and the light indicates with and without methane. So unlike the methane seeps, now we're seeing it in the absence of methane as well. And that really tells us that it's no longer these methanotrophic archaea doing this here, it's somebody else. If you compare that to the rates of ammonium fixed, you can see that in some places where you have more overall activity, you have more nitrogen fixation. But just because you have general activity doesn't mean that you're necessarily going to have nitrogen fixation. Here's an example where you have very high rates of overall activity, but still no nitrogen fixation. So we looked into this a little bit more, and it does seem that the concentrations of ammonium are very important in deciding whether or not you actually fix nitrogen. The trends aren't always dictated by what the in situ concentrations were, but what the concentrations of ammonium are in the actual bottle. And we also measured the concentrations of sulfate reduced, or the rates of sulfate reduced and methane oxidized. You can see here we have a peak in the amount of sulfate reduced and methane oxidized here, which is not where the nitrogen fixation is occurring, which is again telling us that it's different microbes mediating this than what we saw at Seeps. So our next step in this project is to look outside of whale falls at totally regular background sediment. Here we think we might run into an issue with carbon limitation, because like I said, a lot of areas of the seafloor are carbon limited, in which case you wouldn't need to fix nitrogen. But if we do see nitrogen fixation in kind of regular background sediments, that's kind of the area of the seafloor that has the potential to influence the nitrogen cycle the most. So a few months ago now, we collected samples in a transect off the coast of the San Francisco Peninsula. We have set up 15N2 experiments, and we are waiting to see what happens. So I'll have to keep you posted on that. We also collected samples off the coast of Massachusetts asking the same questions. Are we seeing nitrogen fixation in regular background samples? The other thing we're doing is trying to figure out who is doing diazotrophy outside of Seeps. This includes the ones that we saw at the whale fall, because we still don't know who is doing it there. We know it's not just methanotrophic archaea, but we don't know who. So we're doing a few different approaches here: co-occurrence analysis of NIF-H and 16S rRNA genes, and the detection of NIF genes within metagenomic bins that can also link it to 16S sequences. We'll also do density gradient SIP, which is the technique I mentioned at the very beginning, where you extract the DNA and put it in a density gradient tube to separate the unlabeled DNA from the labeled DNA and then sequence all of these fractions. So here, for a particular operational taxonomic unit, which you can kind of think of like a species, You have the distribution of unlabeled DNA in a control sample here, but this one was enriched, so it was heavier when it was exposed to the 15N2. So we're using all of these techniques to create very specific hypotheses that we can then test with fish nanosims, because we can design that specific fish probe to then look at in the nanosims. So all of this is kind of the hypothesis generation stage, so we can get to this point where we answer it definitively. So to summarize the nitrogen fixation part of the talk, there are functional diazotrophs that are widespread in deep sea marine sediments. Diazotrophy in sediments is a significant source of nitrogen locally. So by comparing the amount of ammonium uptake to the amount of 15N2 uptake, we can calculate in some places it contributes up to 30% of their nitrogen requirement. I didn't get into this too much, but we are seeing that ammonium limits deep sea diazotrophy in many places, but because the concentrations of ammonium are low enough in a lot of these sediments, the organisms are blind to it, basically. It's a threshold. It's only if it's above a certain amount that it then has an impact on them. And our future work is looking at whether diazotrophs are limited by carbon availability in regular background sediments, who they are, and ultimately the impact on the nitrogen cycle. Any questions about that part of the talk before I jump into this really different area of the ocean? We can come back to questions at the end, but if anybody wants to ask anything now, I'm happy to answer. Yes?
Could you just say one more time why you suspect the sediments to be carbon limited? Is it because the organic content is too low, or is it because it's refractory?
Good question. It's probably a combination, and probably which one of those two possibilities is ultimately the answer probably varies from place to place, but it's probably a combination.
But isn't it carbon rich, actually?
Like if you were to actually measure the weight % carbon, I guess it depends how you define carbon rich. It's not that high, but if it's inaccessible to microbes, then it doesn't matter anyway what the overall percentage is. So in that case, it is refractory. So it's a combination. Anything else? Yes.
I guess kind of related. If you did find nitrogen fixation in the deep sediments, especially like here off the California coast, like the deep, real deep sediments, do you think there would be a seasonality to it? And like which season do you think you would be more likely to find it in? The low flux season or the high flux season?
I was just talking to somebody about this afternoon. If it is a carbon limitation issue, then there is potentially a seasonality issue where in months where you have more primary production, you would have more carbon deposition to the seafloor, then you would have more potential for nitrogen limitation and more nitrogen fixation. But how affected deep sediments are by seasons is, I think, kind of debatable and questionable, and below a certain depth, the exact threshold of which I'm not sure, I don't think seasonality plays a role at all. That would be something that would be interesting to talk about maybe at the happy hour. Maybe some of you have some opinions on that. Anything else? All right. Our next topic is the activity and metabolic flexibility of marine archaea. So again, looking back at our distribution of microbes through the water column, if you were to map the distribution of archaea to bacteria, you'd find out that it really changes with depth. So at the surface of the ocean, you have very few archaea relative to bacteria, but in some places, in the depths of the ocean, you actually have more archaea than you do bacteria. So who are these archaea? We really didn't know until pretty recently. It was about 20 years ago that this abundance of archaea was even discovered. They are thought to be primarily of a single phylum, the marine group 1 Crenarchaeota, which has been renamed the Taumarchaeota. And they make up about 77% of all of this archaea. In about 2005, the first isolate of this group was obtained, which taught us that at least some of them are capable of chemoautotrophy, so oxidizing ammonia and fixing bicarbonate. In this process, they also produce nitrous oxide, and they're actually the biggest source of nitrous oxide in the oceans. So with such a large group of organisms, to base really large assumptions on a small set of isolates might not be a smart move. So people started to look more at the metagenomic data we had, which is sequencing all of the genes in the ocean to figure out what other flexibility in metabolism they might have. And some interesting evidence suggested that they're also capable of taking up organic carbon. And this really changes their role in the carbon cycle and their role in greenhouse gas production and consumption, because if they're consuming organic matter, they basically play the reverse role in CO2 sequestration. So understanding which of these two they're doing is quite important, in addition to understanding their role in the nitrous oxide production. So the question I wanted to ask is, are Archaea using inorganic or organic carbon in the environment? And there's really good evidence, it turns out, on both sides of this coin already. So the pure and enrichment cultures, as I've already mentioned, have shown that these organisms are capable of autotrophic growth. There's genomic evidence that tells us even what pathways they're using to do this. And there's also stable isotope tracing experiments that have tracked 13C bicarbonate into some of these cells. But there's equally good evidence on the other side from enrichment cultures, genetic data, density gradient, stable isotope probing, marfish, which I haven't explained yet but is similar to fish sims, and radiocarbon, which suggests that at least a fraction of them are taking up organic carbon. So we can conceptually think about this in a few different ways. We either have two populations of these archaea, some of which are autotrophs and some of which are heterotrophs. They could all be mixotrophs, getting some carbon from each of these sources. We could have a combination of all three, or we could have primarily autotrophy with some mixotrophy, and these would explain these kind of seemingly conflicting data sets. To answer this question, I collected samples at Pacifica Pier, which is just off the coast of Pacifica, and off the coast of Los Angeles at the San Pedro Ocean Time Series. To answer this question, are the organisms taking up organic carbon or inorganic carbon, I did stable isotope labeling experiments with two different substrates. So simultaneously incubated with 13C bicarbonate and 15N amino acids, the idea being that organisms could either take up 0, 1, the other, or both of these substrates, and I would be able to measure that, again, with the nanosims. This would tell me if they were autotrophs, mixotrophs, or heterotrophs. And you may have a different exact definition of mixotroph. There are some organisms that you could call an autotroph but still take up amino acids. Here I'm defining them operationally as if it takes up both of these substrates. So we have our nanosims image where each of these little blobs are an individual cell. I don't know who is who in this scenario. I want you to keep an eye on these 4 cells in particular. So right now we're looking at the CN counts. So this is anywhere where there's both carbon and nitrogen. Now we're looking at the 13C-12C ratio. So you can only see the cells that have taken up the 13C bicarbonate. Many fewer cells, right? Of the 4 we were looking at before, only these 2 were capable of taking up that inorganic carbon. Now we're looking at the 15N-14N ratio, and we see a different pattern within these 4 cells. So by screening hundreds of cells like this, you can rapidly get a sense of who's an autotroph, a mixotroph, and a heterotroph, and plot that as percentages. So in this case, we had mostly heterotrophs and some mixotrophs at each site. But we still don't know who is who. So one of the ways I tried to approach this on a bulk level was to add bacteria-specific antibiotics. And when I did this, I looked at the microbial community before adding the antibiotics, where each one of these different groups is represented by a different color. And after antibiotics, I got about 5 times more archaea. I expected this to be even higher. I think there's different ways I could have set up the experiment to do that, but I still got 5 times as much. And that happened at both sites, about 5 times more. So then when I went back to the nanosims, I could see that when I added antibiotics, I had an increase in the number of autotrophs, which is consistent with these archaea being autotrophs. I also had a small increase in the amount of mixotrophy, which suggested potentially mixotrophy too. But I wanted to be more specific, and since we have that capability using fish nanosims, I decided to use a probe specific for the Archaea, which is here shown in green. This is a DAPI cell whose identity I don't know. DAPI stained cell, just staining all DNA. So here you see these two blobs just based on the carbon count, so the cells are there. When we look at the delta 13C and the delta 15N, we can see that the tau Archaea have taken up both the 13C and the 15N, suggesting that it is a mixotroph. In all of these cells that I analyzed, the more 15N that was incorporated, the more 13C was incorporated. So it seemed that all of the cells were taking up these two substrates simultaneously. The question you have to ask, though, is if 15N uptake from amino acids is actually a good proxy for taking up the carbon from the amino acids. So in one study that has looked at this before, where they looked at 10 different groups, when you added doubly labeled amino acids, the more 15N you, so it's doubly labeled here, the more 15N you incorporated, the more 13C you incorporated. So it seemed like that was a good proxy. If you observed the 15N uptake, you could infer a 13C uptake. But when I did the same experiment with my samples, I saw something kind of different. So now we're looking just at tau Archaea, and although there's a few cells that fall along this predicted line, the majority do not. They're assimilating just 15N and not 13C. So we need to think a little bit carefully about how to interpret this experiment. There's really two possibilities. One is that the amino acids are being cleaved by organisms in the incubation that is turning into ammonia and other carbon sources, and the Archaea are just assimilating that nitrogen. So the 15N we see in them is a recycling event. The other option is that they are assimilating the amino acids and they're cleaving it intracellularly, maybe because they want another nitrogen source for their ammonia oxidation pathway. So they could be taking up organic matter more because they want the nitrogen than really because they want the carbon. So either of those two options are possible. So we wanted to do another experiment. This time we collected more water from the San Pedro Ocean Time series, and we did two different things, metagenomics and metatranscriptomics of the water we collected right away. And then we expanded the scope of the substrates that we incubated with to see more specific 13C incorporation. We don't have to worry about recycling as much with the 13C substrates, because if a heterotroph did consume that and then produce 13C labeled bicarbonate, it would be so diluted in the background bicarbonate of the sample that 13C label wouldn't end up being detectable in an autotroph, if that makes sense. So if you see the 13C incorporation, it is a very good indicator of actual heterotrophy or mixotrophy. We focused at 150 meters water depth because we were hoping to get the biggest diversity of Archaea. We did have a large percentage of Archaea, about 20% in the DNA, and it made up a majority of the cDNA. So this was not only an abundant member of the community here, but also a very active one. So my postdoc, Alma Parada, has been working on reassembling these metagenomes. So the idea here is you take all of the DNA from the environmental sample, you break it up into tiny pieces in order to sequence it, and then you try to put it back together into the individual organisms that contributed to it. When we separate all of these different pathways by name, you can see all of the ones that are found in any of the DNA sequences that are attributable to the tau Archaea. And we can also divide these into individual bins. So these are metagenome assembled genomes, theoretically from some small phylogenetically defined group of tau Archaea within the whole population. So we had 46 different bins of tau Archaea, and I'm just showing you the 14 most complete here. But the takeaway is that in each one of these, you can see evidence of both inorganic uptake. So here we have the 3-hydroxypropionate, 4-hydroxybutyrate cycle present in most of these, but then you also have evidence of urea degradation, organic nitrogen transport, organic carbon transport, even within a single one. So they seem to have the genetic capability to take up both organic and inorganic matter, consistent with what we saw at Pacifica Pier when we saw both the bicarbonate and the amino acids assimilated. This is just the results from the DNA. When you look at the transcripts, so what they're actually expressing in situ, you see this same diversity as well. So if there's a star, we detected actual transcripts, and you're still seeing the ability to take up both inorganic and organic matter within individual groups. When we look at the NanoSims data, we can see here is the fish image where our green cell here is the tau Archaea. We can see that it is taking up the 15N from amino acids, but not the 13C. And if I plot all of the cells we analyzed, here's the Archaea on the y-axis in red. Most of them are only taking up the 15N. A few of them are also taking up 13C, but this seems to be primarily about the nitrogen. When we look at different substrates, we see different patterns. So for doubly labeled urea, we see both 15N and 13C uptake. And proteins, we see no incorporation at all. So in these 3 different substrates, we see different patterns in each. When we add substrates that are specifically 13C labeled, and then the 15N is coming from ammonium, which is, again, just a proxy for overall growth, we see no 13C uptake. This particular cell was attached to a bacterial cell, so we can basically throw that out. It wasn't, it could have been getting that 13C from that partnership with the bacteria. We see the same thing for glucose and the same thing for oxaloacetate. So no evidence of organic matter uptake from these carbon sources. To look a little bit closer at the urea results for a second, because I think this could be one of the most interesting takeaways, we see both the 13C and the 15N, which really indicates that they are taking up the urea specifically. People have observed parts of this process before, and the hypothesis is that they are taking up the urea, cleaving it intracellularly with urease, creating ammonia, which they can then oxidize to nitrite for energy. Then they can also take this carbon and turn it into biomass, which is why we see less, but still some incorporation of the 13C. So the fact that we're seeing this 13C assimilation means that this process is really happening intracellularly. They're doing this intentionally as a way to get additional nitrogen for nitrification. We looked in the metagenomes to see if we saw ureases, and we did. So you can see in bold here all of the groups that contained urease from our sample, and if it has stars here, it indicates that that was not just a gene that was present, but it was actually expressed. So this process seems to be happening in the environment in the original sample. So to summarize this part of the talk, from the metagenomics and metatranscriptomics, we saw that the tau Archaea contain genes for organic matter utilization and transcribe them in situ, in addition to those for inorganic assimilation. There's a widespread genetic potential for organic matter utilization, but we really see a different story coming from the stable isotope probing experiments. We're seeing that organic carbon is really the primary, in some cases the sole carbon source. Although some tau Archaea can take up urea and amino acids, our hypothesis at this point is really because they're interested in the nitrogen, not the carbon. We saw no detectable glucose, pyruvate, oxaloacetate, or protein assimilation. And finally, we also saw that the tau Archaea assimilate more nitrogen from ammonia and urea than bacteria, which means they could actually be growing faster than the bacteria, which would be unexpected. It could also be that they're somehow scavenging and storing this N for nitrification, which is a process that hasn't been described before. So if we go back to our physical representation of what's happening, this is my best guess and my understanding about it right now. It seems that most of these organisms are indeed autotrophs. There is some role for mixotrophy in their lifestyle, which is why they have the genes for this organic matter uptake, but they're really primarily interested in this organic matter for its nitrogen. So to step back more broadly and think about the single-cell approach that I've been talking about today, you saw kind of 2 different examples of how you can use this approach. The first was when we were looking at nitrogen fixation in deep-sea sediments. This was a way to test for a specific metabolic function. In this case, it was nitrogen fixation. We could also compare different patterns of activity within phylogenetically similar organisms that were occupying different spatial niches. So whether those sulfate-reducing bacteria were attached to the methanotrophic archaea or whether they were separate, we could separate that out based on this technique. Then we looked at the carbon assimilation in pelagic marine archaea. We could do a high throughput survey to just assess the percentage of cells that were autotrophic or heterotrophic or mixotrophic. And then we could also use multiple isotopes to see simultaneous metabolisms within individual cells, which is really the only way to confirm that what you're seeing is mixotrophy and not two different subpopulations, one of which is autotrophic and one of which is heterotrophic. A few reminders if you're interested in using this technique. NanoSims is limited to observing anabolic metabolism because whatever it is has to be assimilated. It can't observe what's being respired. It's generally low throughput, especially if you're processing the cells with fish ahead of time, so it's best used in combination with data sets that can provide a bigger picture, like you're looking at the entire metagenome in addition. And you do need to be careful about substrate recycling because that can really change the interpretation of your results. So if you're interested in using this technique, you're welcome to get in touch with me. The facility at Stanford is set up as a user facility, so anybody can come bring samples and try to make their own measurement. And I should add here that there's a lot of neat applications of this outside of the microbial world. There's a lot of neat symbioses, there's coral work, so even if you work with bigger animals, you might be interested in this yourself. So I'd like to say thank you to lots of people in my group at Stanford, Jed Furman at the University of Southern California for helping me get the spot samples, my colleagues at Lawrence Livermore National Lab, my previous advisors at Caltech, and Dave Fike, who's my collaborator at WashU, and all of my funding sources. And thank you all for listening.
Substrates to test for heterotrophy, if you fed the same choices you made of acetates or amino acids to the surface bacterial population, do you automatically think that all of those substrates will get taken up?
Just for a general population of bacteria?
Right there, right, that water. Right.
No, it would be some subset and some combination, and some cells probably, even if they had the genetic capability to do it, probably wouldn't do it because it was just a dormant cell.
So because you come to the conclusion that they're not heterotrophic, but the part that I have a hard time understanding, and I don't do this, but it just seems to me like there's an infinite number of biomolecules you could feed bacteria as a possible food source.
So there could be some carbon substrate that they can assimilate that I haven't tested. So that's absolutely a limitation. Fish nanosims is an extremely targeted approach. You have to know the specific species you want to target and the specific molecule you're wondering if they can assimilate. So that's just kind of what we're limited to. It's easier when you're thinking about something like nitrogen fixation where it's just one molecule. So we can add that 15N2, and that's the question. When it's about carbon metabolism more broadly, you're right, we can't add every single thing, so there's that caveat to any conclusion we make. Yes?
Have you done the exercise of taking your nitrogen fixation estimates from the deep sea sediments and trying to see, do you close that gap based on, I mean, you've only done it in a few places so far.
Right. So I think it would be really premature, and I'm really wondering what the results are just of the background sediments before I try to extrapolate. What I have done is looked at the rates of nitrogen fixation I see from the methanotrophs specifically, because there are global rates of methane oxidation attributable to these organisms. So if I take the ratio of nitrogen fixation I see to the amount of methane oxidized and apply that to how much is estimated that they do globally, does that close the gap? And no, it doesn't. It's beyond, it's a small fraction of 1%. So they are definitely not the answer themselves. And I don't know if this will end up being the answer or not. If I were to guess right now, I'd say no. I think the discrepancy has to do with probably an underestimate in a lot of different areas and potentially an overall methodological underestimate that we're all making with our nitrogen fixation measurements. And a few of these have come out in the last few years. A lot of the ways people are measuring nitrogen fixation is to add a bubble of 15N2, and then they do a very short incubation. And now people are starting to understand that it hasn't totally dissolved into the whole incubation. So there's a lot of reasons to think that measurements of nitrogen fixation in all different habitats have been underestimated for different reasons. So I think this might be part of it, but I don't think it's likely to be the entire thing. But, you know, we'll see. Yes?
Are all of these experiments at sea level? You bring these sediments up and they're done on shipboard?
That's right. So one of the things to think about too is all of the ways we've changed their community from their natural experience, and pressure is probably the biggest difference. From that point of view, our rates are probably an underestimate because they're less happy when you take them out of their natural environment than they were when they were at home. Especially with metabolisms involving gas, which are going to be particularly susceptible to partial pressure to different pressures. So I would guess that ours are an underestimate because of that.
And can you say just a word or two about playing with that machine? You know, when you just walk in and push the start button and get an answer, and then shut off the lights and go home, or do you live in that room with that machine?
It's a little bit of a combination. All of the user facilities that have nanosims have dedicated technicians, and so they're the primary person involved with like flying the plane. I almost think of it. They set it up so that that primary ion beam is as well focused as it can be, and you're making sure that you're analyzing the correct masses. Finding the cells ends up being the hardest part of the analysis from the user's point of view because it doesn't have an epifluorescence microscope inside of it. So you have to re-find these particular red or green cells that you found with the microscope now with a very low resolution, basically phase contrast microscope image in the nanosims. So just re-finding them takes a long time. Yes.
So do you think that the autotrophic, all these autotrophic Archaea in the mesopelagic, like how significant of a role do you think they have in like fueling mesopelagic ecosystems?
I have to assume a pretty big one, but I was just talking to somebody else about this, and we haven't seen good rate measurements on exactly what it is quantitatively. So I'd really like to use some of the measurements that I've made to try to make those calculations and extrapolate. One of the things that has limited me from trying to think about that in the past is one thing that I didn't talk about is how much the sample processing for nanosims analysis ends up affecting the isotope ratio. So when we fix the cells with formaldehyde, when we add these oligonucleotide probes with the fluorophores, we are adding natural abundance carbon and nitrogen. So we're basically diluting out that original 15N or 13C signal. So all of our enrichment values are lower than they should be. So if I were to do the calculation now without correcting for that, I would be really underestimating their rate measurements. So what we're doing to try to improve that is use pure cultures to go through the whole process when we know what the original enrichment was, and then process it with all these different things that we're doing, and then measure it again. And with the one species we've done that with so far, it was a 70% dilution. So I think we know that that's an important factor to take into account, and once we know better how to constrain it, I can go back to our actual measurements and then try to extrapolate up into how much bicarbonate they're actually fixing in this region of the ocean and what a fraction of that means for carbon cycling in that ecosystem. Yeah, we're just like a few steps. We're not quite there yet, but that's the direction we'd like to go in. Yeah. Yes?
I guess, could you say something about some of the specific genes that you found that were involved in organic material uptake by the time Archaea? And does that any of those genes, you mentioned marfish as well?
Mm-hmm.
And I don't
that
know,
was Jed's work.
Yeah, and I don't know as well as I should. But the particular molecules that they added, compounds that they added, are they similar to?
So that is actually a really interesting question, because marfish is a technique that relies on radioactive isotopes. And so they were actually using tritiated organic substrates. So if only a fraction of the molecule had been assimilated, like I'm seeing only the nitrogen, that would have registered as, yes, this molecule was assimilated. So a lot of those studies that were saying that that organic carbon was assimilated was actually just confirming that the hydrogen, or really the tritium, was what was assimilated. So that included amino acids, and I think glucose has been done as well. In terms of the genes, we were trying to focus on transporters because there's a lot of genes that are ambiguous, because even an autotroph is going to be reprocessing the organic carbon. So we tried to focus on transporters for both organic carbon and nitrogen, and so that's what the main evidence for their genetic ability to use the organic matter comes from.